the basis of antigen antibody reaction or serological reaction. Fourth one is molecular biological technique, which is also specific or sensitive techniques. And the fifth one, which we have already studied, is cultivation of viruses. So if you see the assay or diagnosis of virus, you will find the property of virus is actually responsible or is one of the important factor for the assay of viruses. यानी कि जो वायरस का ऐसे किया जा रहा है, डायग्नोज किया जा रहा है, वो उसकी प्रॉपर्टी के बेस्ड है. In my initial lectures, when we are discussing about the morphology of the viruses or the components of viruses, when I have compared it with the bacteria, I was trying to explain the things on the basis of its property. हमने बोला कि बैक्टीरिया का cell wall. Why we are studying cell wall? Why we are studying lipopolysaccharide? What is the significance? The property of the specific component organelle present in an organism actually helps you to explore that pathogen. Okay, so in each we can say assay will find its property. तो यहाँ पर कहीं ना कहीं आप उसकी उस property को ही explore कर रहे हैं. So quantal assay, we have discussed that this assay is based on the assay of its infectivity, whether it is infective or not, right? And quantitatively, we determine the number of virus particles. So we determine ID50, which is 50% infectious dose. And according to Reed and Munch method, this is the protocol that we follow in virology. Reed and Munch method, and according to this, the reciprocal of the dilution of the virus that infects 50% of the inoculated host. 50% of the inoculated host means 50% population, irrespective of uh, any of the host. अब host it can be animal हो सकता है, ये cell lines हो सकता है, या वो egg हो सकता है, right? So we determine the infectious dose और its infectivity. यहाँ पर इसका एक एप्लीकेशन वायरस के ऐसे में आएगा जब आपको अननोन वायरस दिया जाए और आपसे पूछा जाए कि वेदर इट इज इन्फेक्टिव और नॉट, वेदर इट इज वर्लुएंट और नॉट, और इन विच सिस्टम इट इज पैथोजेनिक, राइट? सो you will follow the culture method of viruses and you will follow this infectivity. हमने बात की थी LD50, TCID50 और EID50, जो कि लीथल डोज 50 है, जो कि डिटरमाइन करते हैं एनिमल्स में. टिश्यू कल्चर इन्फेक्शियस डोज 50 टिश्यू कल्चर सिस्टम में, सेल कल्चर सिस्टम में, और EID50 एम्ब्रियोनिक एग इन्फेक्शियस डोज 50 विच इज डिटरमाइंड इन एग. और LD50 का ये डेफिनेशन मैंने बताया था, रेसिप्रोकल ऑफ दैट डाइल्यूशन पर यूनिट वॉल्यूम ऑफ वायरस स्टॉक दैट विल किल 50% ऑफ द इनॉकुलेटेड एनिमल्स. यानी कि हम उस डोज को डिटरमाइन करते हैं जो कि 50% ऑफ एनिमल्स को इन्फेक्ट करता हो, किल करता हो, या ऑब्जर्वेशंस आपको शो होती हैं. लाइकवाइज TCID या EID50 कैलकुलेट किया जाता है. So in cut short, if I say quantal assay, this assay is to determine the infectious dose, right? Whether the virus is infective or not. While in quantitative assay हमने बात की थी कि it is an assay in which we determine the number of virus particles, right? Number of virus particles, और इसको हमने लिखा था plague forming unit per ml. ये लिखा है PFU. This is PFU per ml, plague forming unit per ml. So here what you are doing is you are actually assaying the virus, right, on the basis of its load. So if I say कि a sample is given to you, a sample containing virus is given to you, obviously virus must be present, and it is asked to determine the viral load. Viral load. Let us take an example. मैंने आपको एक डॉग का saliva did as a sample, and it is supposed that it is containing virus. So how you will determine the viral load in the saliva? So you will do the quantitative assay. So in this assay, what you will do, you will actually inject the sample, that is saliva, in a suitable host system. So host system, it can be egg, the chorioallantoid membrane, or it can be cell lines, that is cell culture system, right? And you will identify the साइटोपैथिक इफेक्ट, जैसे हमने बोला था प्लैक, जब प्लैक ऐसे में प्लैक तो लोकलाइज्ड एरिया ऑफ वायरस इंड्यूस साइटोपैथोलॉजी, वो क्लियर जोन जो बन रहे हैं उसको हम प्लैक कर रहे हैं, तो यू विल डिटरमाइन अ प्लैक। यहाँ पर मैंने एक एग्जांपल दिया था व्हेन हाउ वी डिटरमाइन द बैक्टीरियल लोड इन ए गिवन सॉइल सैंपल, एक एग्जांपल दिया था। लाइकवाइज सेम you do the virus dilution, you identify the cytopathic effect, and which is called as plaque. So plaque number is 35 into reciprocal of dilution upon inoculum taken, and this is how you will determine the plaque forming unit per ml. So this number one quantal assay and quantitative assay. Next I am taking, next was the physiochemical properties of viruses, यानी कि हम वायरस को डिटरमाइन करेंगे, ऐसे करेंगे ऑन द बेसिस ऑफ इट्स physiochemical properties. Now let us take it as an example. First I am writing a viral assay on the basis of physiochemical properties. Okay, let us suppose take the theoretical knowledge what you have gained till date is the composition of virus. कंपोजिशन ऑफ वायरस, आपको अभी तक थ्योरेटिकल ये पता है कंपोजिशन ऑफ वायरस, और आपको पता है द मॉर्फोलॉजी ऑफ वायरस, वायरल मॉर्फोलॉजी। ओके, now let us take an example. Maybe you are not able to get the point, maybe you have not studied, but let us take an example. Say for example I have given you a sample containing this sample containing bacteria, yeast, virus. A sample is containing bacteria, yeast, and virus. It may be mixed sample, it may be a separate sample, but you don't know in which sample the bacteria is present, or in which sample yeast is present, or in which sample virus is present. And it is asked that identify the organism on the basis of its morphology. On the basis of morphology, can anyone tell me how you will identify these organism, or how you will differentiate this organism on the basis of its morphology? मैं अगेन क्वेश्चन रिपीट कर रहा हूँ। क्वेश्चन ये है, you know the morphology of the organism, you have an idea of the morphology of at least virus. It is asked that a sample is containing bacteria, yeast, and virus is given to you. How you will identify or how you will differentiate these organisms on the basis of its morphology? How you will do this test? Can anyone tell me? या कोई भी कुछ इसके बारे में आइडिया दे सकता है, गेस कर सकता है?
So according to the shape and size.
Okay, so when we say morphology, absolutely right, we say it is shape and size. Okay, so we know the shape. So where you will identify this shape and size, and how you will differentiate? आप किस इंस्ट्रूमेंट को अब यूज करेंगे?
So microscope, through microscope.
Microscope, absolutely. So we say that okay, we can use microscope, and since we know the size of a microorganism, we will identify these organisms on the basis of its size as well as shape as well as arrangement. So shape, size, and arrangement. Okay, so let us suppose if you are using a bright field microscope or oil immersion microscope, and at 100x this is objective lens, or total is say 1000, right? This you can differentiate bacteria and yeast. So if you say bacteria is observing like this, say if it is a rod shape, or if it is a circular foci, you will identify this one, and if it is yeast, it will observe like this way, Okay, so if it is unknown to you, the sample is unknown to you, and even if it is containing cocci and here it is yeast, you can differentiate the organism on the basis of size. So this is bacteria, ये bacteria है and ये yeast है. So theoretically you know the size of the organism and you can easily differentiate. Same if it is asked, just confirm whether the sample is containing virus or not. So you can identify the viruses under electron microscope, isn't it? And using electron microscope you can identify the size or shape of virus. So this is a preliminary assay of virus, we can say, identification of virus on the basis of which property? On the basis of its size and shape property, because you know the size and shape of the virus, so you can confirm this by electron microscopy. So the answer, the first answer is the physiochemical property is identification of size and shape of virus by using electron microscope. So यहाँ पर property जो आपकी डिटरमाइन कर रही है वो उसकी मॉर्फोलॉजिकल प्रॉपर्टी है, राइट, दैट इज इलेक्ट्रॉन माइक्रोस्कोप आप यूज कर रहे हैं और इसकी प्रॉपर्टी आप यूज कर रहे हैं मॉर्फोलॉजिकल प्रॉपर्टी. नाउ लेट्स टेक second identification. Let us suppose the given virus sample is having envelope or may not be have any envelope. So you are trying to assay the virus, assay of virus as whether it is enveloped or naked. Fine. आपने जब मॉर्फोलॉजी पढ़ी थी तो आपको पता चला था कि वायरस में भी एनवेलप्ड, वायरस में भी नेकेड. And this enveloped viruses may contain different type of envelopes. Nature of envelope varies and it may be made up of lipids or other lipid components. So if again I am giving you a problem first so that you can identify. Let us suppose viral sample is given to you, okay, and it is asked to identify the virus whether it is enveloped or naked. Okay, I am here taking example enveloped as this your lipid envelope. Okay, simple question is a viral sample is provided to you. It is asked to assay the virus, to confirm the virus whether it is enveloped virus or naked virus. I am taking example of enveloped virus as lipid containing enveloped virus. Can you guess how you can identify this? So what are the property which is responsible for this assay? यानी इस वायरस की assay या diagnose करने में उसकी कौन सी property है? पहला, the chemical composition of the virus. The chemical composition means a typical virus, it may be enveloped or it may be naked. So if it is enveloped, then how you will identify? Or if it is naked, then how you will identify? So it may be either enveloped or naked. How you will confirm it? यहाँ पर दो प्रॉपर्टी मैं आपको यहाँ पर लिख देता हूँ कि यू आर डूइंग दिस एसे ऑफ द वायरस ऑन द बेसिस ऑफ वायरल एसे. ये सिर्फ हम अपने समझने के लिए लिख रहे हैं कि वायरल एसे ऑन द बेसिस ऑफ फर्स्ट viral composition. I am giving a hint actually. Viral composition, that is chemical composition, and second is culture characteristics on cell lines or the growth of virus on the cell lines. यहाँ पर दो properties आप use कर रहे हैं. First is the virus grows on the cell lines right, and show cytopathic effect. And second property, it has specific composition and it may be enveloped or it may be naked. Now, can you guess how you will identify this? Because in quantitative examination, the question can be asked on this basis to determine the property of the virus. So the basic introductory knowledge will help you to understand the points. कोई guess कर सकता है कि इन दो prop basis पे यानी composition और culture characteristics पे how you will determine whether it is enveloped or naked? जहाँ पर आपको अभी तक इतना पता है कि virus may be enveloped, may be naked, and if you infect the virus in cell lines, it will show cytopathic effect. If it is virus, if it is virulent, if it is active, it will show a cytopathic effect. Yes, anyone, any student can guess. Guess करो, no problem. I have no problem. जिसको जो समझ में आ रहा बोलता है. Question is clear to you? Question is clear to you now? Yes or no?
Yes,
Yes,
sir.
sir.
Okay. Let me give you the example how you will. I am trying to sensitize you to explore the reasoning approach. Otherwise, what will happen? You will just simply learn the things, right? But you cannot recall for a long time. So मैं यहाँ पर आपको reasoning approach के साथ बता रहा हूँ कि why, how we are doing the viral assay, है ना? हम यहाँ पर we know actually the basic concept and we are applying it for the diagnosis of the virus sample. Okay, now let us suppose take an example. A sample, virus sample is given to you. So I am giving an equation, virus, this is sample. I don't know whether it is enveloped or it is naked. But if I know that the property of this enveloped virus is that it is sensitive to, can anyone tell? Filling the blanks. Enveloped viruses are sensitive to
Sir, heat.
heat, okay, और कोई? मैं इस क्वेश्चन को पिक कर रहा हूं आपके उस पॉइंट से जहां पर हमने पढ़ा था फिजियोकेमिकल प्रॉपर्टीज ऑफ इट अपलोड किए जरूर उसको पढ़ते रहिए. अगर ये एनवलप वायरस है और मैं हिंट दे रहा हूं लिपिड वायरस, इट मींस इट इज सेंसिटिव टू ऑर्गेनिक सॉल्वेंट्स. इट इज सेंसिटिव टू ऑर्गेनिक सॉल्वेंट्स. ऑर्गेनिक सॉल्वेंट्स लाइक क्लोरोफॉर्म या ईथर. Fine. I have given you the one statement that the one of the it is sensitive to organic solvents, that is it is actually inhibited by organic solvents. So what we are doing, we are incubating this virus sample with organic solvent like chloroform and ether. Am I audible? Just to confirm, I am audible now?
Yes, sir.
Yes, sir.
ओके फाइन, ठीक है. सो हमने क्या किया कि वायरस सैंपल को ऑर्गेनिक सॉल्वेंट दैट इज क्लोरोफॉर्म ईथर के साथ मिक्स किया. ओके, एंड देन आई एम इन्फेक्टिंग इट इनटू द सेल कल्चर. आपको कोई नई चीज नहीं बता रहा हूं मैं, आपने अभी तक जितना पढ़ा है उसी को अप्लाई कर रहे हैं. सो आई एम इन्फेक्टिंग इट इनटू द सेल कल्चर. व्हाट आई एम डूइंग इज फर्स्टली आई विल मिक्स द वायरस सैंपल विद द ऑर्गेनिक सॉल्वेंट, right, and then if infecting it into the cell culture. जो आपने cultivation पढ़ा था वही पर कर रहे हैं. Now what will happen? There are two possibilities. One, option observation of cytopathic effect, which confirms virus cytopathic effect, which confirms viral growth. Okay, and second possibility is no cytopathic effect, that is no viral growth. Okay, so after incubating it with using with suitable solvent and infecting into the cell culture system, there may be two possibility, because till this time you don't know anything about the chemical composition of the virus. So either you will observe cytopathic effect that will confirm viral growth, or you will observe no cytopathic effect that will confirm no viral growth. Now, can you tell me in which case it is enveloped and in which case it is naked? I am labeling it as observation A and observation B. Now, can you tell me in which observation it is enveloped and in which observation it is naked?
Sir, A is envelope.
A is
Envelope.
enveloped, okay, and B is naked,
Naked.
obvious, okay. और कोई student? I am just again showing you this
सर
thing.
जो साइटो सर जो ए है उसमें ही दोनों होने चाहिए ना, बिकॉज जो बी है उसमें तो सर वायरल ग्रोथ ही नहीं है, तो
ग्रोथ
अगर
नहीं
नेकेड
है,
भी होगा तो फिर ग्रोथ कैसे शो करेगा वो?
ओके, तो आपके हिसाब से व्हाट इज द राइट आंसर?
सर ए में मतलब दोनों होने चाहिए, लाइक एनवेलप्ड एंड नेकेड।
ओके, आपके हिसाब से एनवेलप्ड एंड नेकेड। मैं नाम भी पूछ सकता हूँ, पहले कौन सा स्टूडेंट था जिसने
Sir,
एनवेलप बताया
Shweta.
था? श्वेता। ओके,
Yes, sir,
so
Shweta.
I will first I will appreciate you for giving this answer actually to what, and second जिसने दोनों दिखाया?
सर वृंदा।
वृंदा, ओके वृंदा। और and in this case B, here
सर
it
बी में तो अगर वायरल ग्रोथ ही नहीं है तो फिर मतलब लाइक नो वायरस।
no virus, okay, so no virus, okay, no virus, fine. Other students, boys, I am again showing you the equation. Virus,
So,
yeah, yes.
so I think A is non-enveloped and P is enveloped.
So according to you, क्या नाम बताया आपने?
Ragini.
So according to you, this A observation is basically naked and B observation is enveloped, isn't it?
Yes. Yes, sir.
Okay, yes, yes, yes. Okay. और कोई wants to give this answer? मैं इसलिए ऐसा कर रहा हूँ so that you can recall it for a longer time. आपको लंबे समय तक पॉइंट्स याद रहेगा कि हाउ वी कंफर्म दिस थिंग। और कोई स्टूडेंट? ओके, देन ओके, मुझे रागिनी कंफर्म करेगी कि व्हाई अकॉर्डिंग टू इट इज एनवेलप्ड।
Because तो सेल का लाइसिस हो रहा है, मतलब उसमें हमें शो हो रहा है, लाइसिस हो रहा है, उसमें साइटोपैथिक इफेक्ट शो हो रहा है हमें।
यस यस
वेयर इज इन
यस।
एनवेलप्ड हमें उसमें साइटोपैथिक इफेक्ट हमें शो नहीं होगा।
ओके, क्यों?
तो बिकॉज उसमें लाइसिस नहीं हो पा रहा है।
क्यों?
तो
तुम प्राइस तो जीत गई हो, बट आंसर तुम्हें सही बताना होगा। Yes, this is
yes sir,
absolutely right. Yes, this is absolutely right answer, Ragini. Answer
yes
सही
sir.
है। Now let us see what is the point. I know that the one of the physiochemical property of virus is the enveloped viruses are sensitive to organic solvent. इसके पीछे principle है, it is sensitive to organic solvents. What does it mean? इसका मतलब क्या हुआ? Sensitive to organic solvent. It means enveloped viruses are inhibited by organic solvents and are not able to infect or multiply or show cytopathic effect. Yani if you mean, it's, we can say it's like a sterilization method or control of viruses, we can say, because of this is the property. So enveloped viruses, which is lipid containing, it is inhibited by organic solvent. What we have done is we have mixed the virus sample with organic solvent. Now assume that it is not a enveloped virus. मान लेते कि वो enveloped virus नहीं है, तो क्या होगा? If it is not an enveloped virus, it is naked virus, then there is no effect of this organic solvent on the virus, and virus will remain infected. And if it is effective, it will show cytopathic effect, right? Because it is not inhibited by organic solvent, and in this case it will show cytopathic effect. तो naked envelope जो होगा वो cytopathic effect show कर रहा होगा. But let us assume that it is a enveloped virus, lipid containing enveloped virus. Then what will happen? It will inhibited by this organic solvent, and since it is inhibited, when we transfer it to the cell lines, it will show no cytopathic effect. And if no cytopathic effect observed, it means it is a enveloped virus. अगर ये enveloped होगा तभी तो cytopathic effect show नहीं कर पा रहा है, because it is inhibited by organic solvents. So using organic solvents, you can differentiate or you can assay the virus whether it is naked or enveloped. तो आपने enveloped और naked virus को identify कैसे किया? On the basis of this property. One of the property that the enveloped viruses are sensitive to organic solvent. And this gives you an idea कि okay, if what is the nature of the envelope, and accordingly you can treat it with suitable inhibitor, right? So that you can confirm whether it is enveloped or naked. You got the point? समझ में आ गया?
Yes, sir.
अब इसमें एक इम्पॉर्टेंट एस्पेक्ट क्या है क्वेश्चन में? क्वेश्चन को आप पढ़ेंगे तो दो स्टेटमेंट है. पहला स्टेटमेंट है कि वायरस इज incubated or mixed with organic solvent followed by infection on cell lines. दो स्टेटमेंट है. इसमें ये नहीं कहा गया था कि वायरस इज इन्फेक्टेड इनटू द सेल लाइंस डायरेक्टली. अगर ऐसा होता तो मुश्किल था कंफर्म कर पाना. So virus is not infected into the cell lines directly. It is first mixed with organic solvent. So hoping that you have got this point. Again taken a same example and we'll explore the other property of this virus on the same point. Now let us suppose a virus sample is given to you, okay, and since you know that the virus contain either DNA or RNA, okay, so virus sample is given to you. It is asked to confirm whether it is DNA containing virus or RNA containing virus. So the heading is identification of nucleic acid type. Assay of viruses on the basis of its chemical again composition, that we are determining its nucleic acid. Identification of virus on the virus by nucleic acid type, whether it is DNA containing or RNA containing. Now it is open to all. Just guess what can we do, not specifically, but what will be the idea for doing this experiment? How we will identify this thing? How you will identify whether it is DNA containing or RNA containing virus? Yes. Concept is same. Concept is same as in earlier case. Again, the concept is same. Again, you have to mix it with something and then infect into the cell lines and then identify whether it is growing or not. That is same. Now, can you guess? It will open your reasoning approach actually. That's why I'm not giving you the answer, or I'm not giving the Any student? Quickly guess the answer. In this case, what we have used? In this case, we have used the inhibitor. Here we have used organic solvent as an inhibitor, and it will inhibit the envelope. Now I've given you the hint actually. Now what we can do? Let me write the equation. Virus plus what you will take here. वायरस को आप किसके साथ मिक्स करेंगे, ट्रीट करेंगे, क्या हो सकता है?
Sir, I think nuclease enzyme.
Protein.
Okay, absolutely right. It means approach is absolutely right. Since we are trying to identify the nucleic acid, so obviously either we will take any of the inhibitor which will either inhibit DNA or RNA. Getting my point? आपको पता करना है कि whether it is DNA containing or RNA containing, so you will take inhibitor that will specifically inhibit either DNA or RNA. किसी एक को, दोनों को inhibit करेगा, then you are not able to identify the nucleic acid type. So you will take inhibitor. Here I am taking an example which is very commonly used in laboratory is BUDR. BUDR stands for 5-bromo 2-deoxyuridine. 5-bromo-deoxyuridine, which is a inhibitor of DNA synthesis. DNA synthesis. So it inhibits the synthesis of DNA. So maybe you know the DNA, that is nucleic acid or adenine, guanine, thymine, cytosine. They are synthesized by two pathways: de novo pathway and salvage pathway. So if we use a inhibitor which inhibit the nucleic acid synthesis, that is DNA synthesis, now what will happen? So here what you have done, you have taken an inhibitor which will inhibit DNA, but you don't know whether the virus is DNA containing and RNA containing. The same. Here what I have written in the first case, I have written okay, we have written cytopathic effect observed. So again I am writing the same equation here. Virus is mixed with inhibitor. Here it is 5-bromo 2-deoxyuridine, which is a DNA synthesis inhibitor, and in this case observation A and observation B, just to differentiate. Here cytopathic effect observed, no cytopathic effect. Now can you tell me, in case of observation A, what is the nature of the virus, and in case of observation B, what is the nature of the virus on the basis of its nucleic acid? Yes.
अच्छा, ये इनहिबिटर जो है वो सेम होगा दोनों के लिए, डीएनए और आरएनए के लिए, या फिर डिफरेंट डिफरेंट होंगे?
I have used BUDR, which is a DNA synthesis inhibitor.
Okay, sir.
Okay, I have used inhibitor which will inhibit DNA synthesis. And in observation A, CP is observed. In B, no CP observed. We are assuming that there may be two kind of possibilities. So if CP is observed, then what does it mean on the basis of nucleic acid? Whether it is DNA containing or RNA containing, that is the question. इस केस में DNA containing होगा या RNA containing होगा? Common sense, nothing rocket science. Cytopathic effect is observed. So whether it is DNA containing virus or RNA containing virus. Virus is mixed with an inhibitor which will specifically inhibiting the DNA synthesis. Since we don't know virus is containing DNA or RNA, but we are mixing it with the inhibitor, we are treating with the inhibitor followed by infection in the cell lines. ये मैंने यहाँ पे लिखा नहीं है, but understood है कि you will infect the cell lines. Okay, infect cell lines. After infecting and after incubation, ये मैं default लिख नहीं रहा हूँ because you know the cultivation of virus in cell lines. That's why I'm not writing here. But it is obviously you will infect and you will incubate and you are observing cytopathic effect. If cytopathic effect is observed, then what does it mean? Is it DNA containing or RNA containing? Very quick, PG students. Shekhar.
Yes, sir, Shweta. Shweta Shekhar.
Okay. Yes.
Sir, I think जिसमें इफेक्ट नहीं होगा वो डीएनए होगा.
जिसमें
नो,
cytopathic
जिसमें साइटोपैथिक इफेक्ट नहीं शो होगा होगा.
उसमें वो क्या होगा?
सर डीएनए.
Okay, and this will be RNA. So other students, is it okay? आप फेवर में है इस आंसर के? Yes, the answer is absolutely right. So what we are saying is this BUDR is a DNA synthesis inhibitor. Let us assume that the virus is DNA containing. So what will happen? It will inhibit the DNA synthesis, virus is not able to multiply and hence no cytopathic effect will observe. No observation. Remember, no observation is confirming that it is a DNA containing virus. But if it is RNA containing virus, it will be not inhibited by BUDR and hence CPE will observe. So in case of CPE, it is RNA containing virus. Okay, it may happen practically while doing the experiment. If it is DNA containing virus, it you may observe cytopathic effect but very less CPE, less growth compared to the control one. Remember one point, always you will take a control plate, control flask, always, in every case. Control means not treated, right? Not treated, because you, since in this case you are mixing this sample with the inhibitor, so you will take the control flask, and in case of control flask obviously cytopathic effect will observe, so you can compare it with this. So in this case you will find very less CPE because maybe the inhibitor will inhibit the DNA synthesis but some virus may able to grow, तो virus cytopathic effect आपको बहुत कम दिखाई देगा. Control में there will be very clear cytopathic effect, and in this case also the cytopathic will be observed. So this is how you will identify the nucleic acid type. So this is the assay of viruses on the basis of its nucleic acid type. Okay. We have discussed one of the property that some viruses can agglutinate RBCs. Okay. Virus sample is given to you. Okay, it is asked whether it is containing HA, hemagglutinin, or no HA, no hemagglutinin. आपने एक प्रॉपर्टी पढ़ी थी, इफेक्ट ऑफ वायरसेस ऑन सेल्स, लाइटिक इफेक्ट, लाइसोजेनिक इफेक्ट, हीमा एब्जॉर्प्शन और एग्लूटिनेशन और किसी और transformation effect. So virus sample is given to you. It is asked confirm whether it is containing hemagglutinin or not. Right? The proteins present on the surface of the virus. How you will confirm? Viral surface proteins is known as hemagglutinin. NINS hemagglutinin. जो लेक्चर्स लिए गए हैं आंसर सब उसी से कोरिलेटेड है. ऐसा नहीं एक लेक्चर हो गया दूसरा लेक्चर हो गया देयर इज नो कोरिलेशन. कोरिलेशन इज बिटवीन इवन कोर्सेज इवन. So one course may be correlated with the other course. So it is asked whether it is containing hemagglutinin protein or not. How you will confirm? Give me the answer quickly.
सर हम कोई इनहिबिटर लेंगे जो प्रोटीन को रेगुलेट करेगा.
Okay, but in this case, how you will identify whether it is hemagglutinin? अगर वो inhibit कर देगा तो वो इस condition में आ गया ना, नो एच ए. यानी नो एच ए और द वायरस विच इज कंटेनिंग हीमोग्लोबिन इसको अपने इनहिबिटर से ट्रीट कर दिया, वो भी नो एच ए कंडीशन में आ गया. तो हाउ यू विल डिफरेंशिएट? आपको कंफर्म करना है वेदर द वायरस इज कंटेनिंग दिस वायरल प्रोटीन्स और नॉट. हर बात नहीं इनहिबिटर लेंगे. कॉम्पिटिटिव क्वेश्चन में क्वेश्चन कॉम्प्लेक्स भी हो सकते हैं और बहुत सिंपल भी हो सकते हैं. जो आपको लगे ना कि इतना भी सिंपल क्वेश्चन पूछा जा सकता है. हियर द केसेस, यहाँ पे वही केस है. द आंसर इज वेरी सिंपल. You have to just apply the knowledge of this property of virus that it when मैं आंसर और नहीं बताऊंगा. Yes, hemagglutination or hemaabsorption. Hemagglutination, I am using the word, which is one of the property of the virus. Quickly, let me write the equation. Virus plus, now what I will fill in the blanks.
So it will bind to the infected cell.
Will infect the cells.
They bind to the infected cell.
Okay. But point is कि this no HA is able to infect the cell lines, is able to grow the virus and grow. This will also able to grow. You have to confirm whether this virus is containing hemagglutinin or not. That's it. आप वायरस को किससे मिक्स करेंगे? If you are not able to give the answer, open the lecture जहाँ पर आपको the effect of viruses on cells बताया गया था. बहुत सिंपल आंसर है. What will do? I will mix this virus with red blood cells. हीमा मतलब क्या होता है? हीमा मतलब blood, right? Here it is red blood cells. Hemagglutination याद है? लैक्टिक इफेक्ट, लाइसोजेनिक इफेक्ट, ट्रांसफॉर्मेशन इफेक्ट और हीमाग्लूटिनेशन एब्जॉर्प्शन. जहाँ मैंने बोला था इट विल एग्लूटिनेट आरबीसी. सम ऑफ द वायरसेस can agglutinate RBCs. So what you will do, you will mix the virus with RBC. What will happen? Either hemagglutination happens or no HA, no hemagglutination. Simple. So if hemagglutination occurs, it means it is the virus is containing hemagglutinin, and if no hemagglutination occurs, it means it is not containing hemagglutinin protein. Okay, confirmed. Now this question be asked in two ways in competitive examination. That take the example. First is, as virus sample is given to you, how you will differentiate the virus on the basis of the viral protein, that is hemagglutinin, right? आंसर दिए हुए हैं, हीमाग्लूटिनेशन एटसेट्रा एटसेट्रा। इस क्वेश्चन में बोला गया है कि वायरस, आपको पता ही नहीं है कि वो वायरस हीमाग्लूटिन कैरी कर रहा है नहीं कर रहा है, एक क्वेश्चन। दूसरा क्वेश्चन, ए सैंपल इज गिवन टू यू, sample is given to you suspected to have viral proteins। एक सैंपल दिया गया है और आपको कंफर्म करना है कि वायरस प्रेजेंट है या नहीं है, प्रेजेंटेड, वायरस प्रेजेंट है या नहीं है, बट तो जो वायरस सैंपल है वो वायरल प्रोटीन या हीमोग्लूटिन कैरी कर रहा है। यानी जो सैंपल जहां से लिया गया है उसके सिम्टम और साइन बेसिस पे इट इज कंफर्म दैट इट इज ए हीमोग्लूटिन containing protein, just confirm whether it is having virus or not। क्या वायरस है या नहीं है, सिंपल ये पूछा गया है। सेम केस, what you will do, you will mix it with RBC। in both case you will mix it with RBC। if HA occurs and no HA occurs, no hemagglutination, if HA occurs then you say virus is present, and here virus is not present। A virus sample is given to you containing hemagglutinin protein, which of the following statement is true? It will bind with RBC, hemagglutinin happens, it will bind with RBC, no hemagglutinin happens, no effect on RBC, none of the above। So when I'm saying viral proteins, that is hemagglutinin protein, always it will bind with RBC and show a agglutination reaction and confirm virus is present। तो यहाँ पर हमने observation क्या है, virus is present। यहाँ पर observation क्या है, whether it is containing hemagglutinin or not। Same experiment, same concept, but the observation on the basis of query is different। Here the answer is yeah, it is containing hemagglutinin, or here it is confirming whether the virus is present or not। Okay, so today we have discussed about the identification of virus or assay of virus on the basis of its physiochemical properties। That was the second point, this one, physiochemical properties। Then next lecture we'll discuss on the basis of serological reaction, and I will suggest you to kindly read the immunology book. You can even refer Tortura as well for basic antigen antibody idea, about basic knowledge of about antigen antibody। Maybe you don't know about antigen antibody, and also suggest you to ask the questions about this immunological reactions or test while at it, because I am applying the knowledge of immunology, that is antigen antibody reaction, for the identification of viruses। So that's it, that's why I'm asking this thing। So please go through it, and if you have any query you can ask me। So in today lecture, if you have any query, it is open। I am again opening this slide। if you have any query you can ask me, कोई भी question में doubt है you can ask me। So first was on the basis of its size and shape, electron microscope. Second was whether it is enveloped or naked, whether it is DNA containing or RNA containing, whether it is hemagglutinin protein containing or not containing. That is one. So any doubt, any query, can ask. Any query? You can write your query in the chat box or conversation box. Yeah, any query from any student. Don't feel shy or don't hesitate. If you have any query, you can ask me. So is it okay? Shall I end the meeting?
Yes, sir.
Okay.
Thank you, sir.
Thank you, sir.